Hello, my name is Tolu from the Kawe team, and today is a first time we're actually doing a live stream study session for Kawe. So, Kawe is a study assistant platform where you can create a study plan leading up to your exam. Many more of these. To those that maybe like to do study with me is online. So, this is the first study session that we're going to do. So I'm just going to walk you through it and we'll do a study session together. So here I'm going to click get started. And here I'm already logged in. So I'm just going to create a study session and a study plan. So let's get into it. I'm going to choose a course. I'm going to try to do neuroscience. So let me try to figure out. I graduated from engineer now, so it's been a while since I've taken neuroscience. So I'm just gonna try to look through my old material and go through neuroscience again because I really like neuroscience. So I'm looking forward to studying it again through this study session. So I'm going to go through that with you. So I'm just really looking up a few things here on my end, and then we can get started. All right, so I'm just going to put neuroscience. So here I'm going to choose when my exam is. So I'm just going to say, let's just say January 5th. Now I'm going to put in some topics that relate to my neuroscience exam. So here, neuronal structure, neurotransmitters and receptors, neuroanatomy, sensory systems. Let me do that one again. Let's see, motor systems, learning and memory, neurodevelopment. So these topics would just be classes that relate to your exam. So here I'm just putting topics that might be on a neuroscience exam. So here I'm just putting topics that relate to neuroscience. So in your case, you would probably take these topics off of your syllabus and you'd put them here. So I'm just gonna look to say favorite time. Let's say morning. And then I'm going to click create schedule. So when you click create schedule, it takes, depending on how long your exam is how long it usually takes. So right now it's creating the schedule for you based off of the topics that you're taking. So that's what I'm doing now. So, yep, Kawe was launched around publicly in October. So we've just built a public version at the end of this last quarter or so. So now we're just looking for people to try it out and use it to study for their exams, for their finals, and go through a unique way of studying, which is creating a study plan and then doing a study session. So really excited for people to try out Kawe and very excited to do a live stream today of our first live stream really of a study session. So super excited. So right now it does take a bit of time. We're trying to find ways to make it a bit faster. maybe you might want to refresh or something a bit faster. But here you can see it's created my study sessions. So for each day leading up to my exam, it's created a study session. So one thing is, and we're still working on, is the time. So I know 3 to 5 a.m. is not really the most ideal time to study. So you can edit the times and making it better. So basically for every day leading up to the exam, we have topics and a description for each topic. So for today's study session, I'm gonna switch the time here. What time is it? Let's just say 8 to 9 a.m. So I'm gonna save my. Fortunately, yeah, you would have to change it for each one of them, but looking at making it much more streamlined in the future. So let's see, I'm gonna just do some of these, but really the time you do it doesn't really matter on the app. You can do a study session at any, changing the time for the first 3 or 4 of them. So once you finish, you'd click continue here. So once you click the dashboard to show you how many days you have until your exam. So here is a great way to keep track of your progress leading up to your exam. And it's a great way to use it as a reminder, because when I was in school, procrastination definitely did come up every now and then. So sometimes you study 3 days before the exam, 4 days before the exam. I started studying right away. So now we can go into a study session. So here we have our first one, neuron, to go to it. So here it's saying it when it's starting. So it's starting in 11 minutes. So we can join it now. And before I go into that, I'm actually going to show you of study sessions. So here I have some that I did relating to software engineering, specifically front end. So I did some study sessions for that, just to review topics within front end. So did some of those, and then I have some here that are in progress and missed. And for any of these topics, like for example, I did immunology, you can always go back and basically do the session. And I'll show you what sessions are later on. But here you can see I have all my neuroscience topics right here. So here I'm going to just go back, click join session, and click start session, and start a study session. Study session for neurology, so let's just get started.
All right, Ninja Nerds, in this video today, we're going to talk about the structure and function of neurons. All right, guys, before you guys get started in this video, please hit that like button, comment down in the comment section, and please subscribe. Also, down in the description box, we'll have links to all our social media platforms for you guys to interact with us. All right, Ninja Nerds, let's get started. All right, Ninja Nerds, so the first thing that we have to talk about when we're talking about a neuron is obviously go over the different structural components of a neuron. So what makes up a neuron structurally, then we got to talk about what those different components of a neuron do. All right, so first things first. When you take a look at this neuron, you see these here, all these little extensions are called dendrites. That's the first thing I want you to know. So the extensions that are coming off of this neuron, receptive zone. We'll talk about what that means for a neuron. Next thing, you have this big circular structure here with a whole bunch of stuff inside of it. This here is called your cell body or your soma. So this is called the cell body, or also sometimes referred to as the soma. Long elongated portion here that's going to be in between the cell body and the axon terminal. Okay, the part here is called your axon. Now I have to add in one more little subcomponent of the axon. It's important because it's going to come up when we talk about action potentials. The part where the cell body kind of like narrows and goes into this kind of like thin axon structure is called the axon hillock. So remember, whenever you're looking at an axon like this, if I were to draw another small version of it like this, there's a part where the cell body starts to narrow. That portion where the cell body kind of like narrows like a funnel, this portion here is referred to as the axon hillock. Okay, so when you're talking about the axon, a special part to remember is called the axon. Important is because there's a high concentration of voltage-gated sodium channels there. So whenever action potentials are generated, they're generated here and move down the axon. Bulbous-like structure here of the neuron coming off the axon. And this is called the axon terminal or the synaptic. And sometimes you might even hear it written as axon terminal bulb or synaptic bulb. There's a bunch of different synonymous terms there, okay? Talk about what their functions are first. All right, so now let's go start talking about the functions of these different areas of the neuron, right? So when we talk about the dendrite, at one of these dendrites here coming off, right? And we're kind of gonna cut into this, right? And really zoom in on it in this view. So we're taking this dendrite, taking a section of that cell membrane and looking at it here. Now, another thing to add on to that is I want you to imagine, this is what we're going to call our postsynaptic neuron. That means that there's going to be other neurons that are synapsing on this postsynaptic neuron. So let's imagine here that you have other neurons interacting at that site that we're going to zoom in on. When we zoom in on that, we got some proteins that you guys need to know because they're relevant to the function of the dendrite. In this dendritic cell membrane part, you have these special types of channels. What on the dendrites are called your ligand-gated ion channels. And these are important because they are involved in formation of EPSPs and IPSPs. What the heck is that, Zach? I'll explain to you what that means. These neurons, right? Imagine here I have kind of like a little neuron here, like a little axonic extension, and it's releasing out a neurotransmitter, okay? that neurotransmitter, what it's going to do is it's going to come here and bind into this little pocket, okay, on this ligand-gated ion channel. When it binds onto the pocket, there's normally like a little valve, if you will, that's blocking this opening for ions to come in, like this. But once this little neurotransmitter binds onto that pocket, it causes that valve, if you will, to open up. When it it's released in here. I'll explain to you what that means. These neurons, right, imagine here I have, and it's releasing out a neurotransmitter, okay? That neurotransmitter, what it's going to do is it's going to come here and bind into this little pocket, okay, on this ligand-gated ion channel. When it binds onto the pocket, there's normally like a little valve, if you will, that's blocking this opening for ions to come in, like this. But once this little neurotransmitter binds onto that pocket, it causes that valve, if you will, to open up, and then allows for positive ions to move into the cell, making the cell nice and positive. That's called depolarization, when you make the cell more positive or less negative, if you will. And that is referred to as a depolarization, right? When you make it more positive, and that's called an EPSP. You're trying to stimulate the neuron to fire or generate an action potential. And the other aspect, let's say that you have another neuron over here. Maybe it's this one. And you have the axon here. And this axon is releasing another neurotransmitter. But instead of this neurotransmitter being stimulatory, let's have the opposing. Let's have an inhibitory neurotransmitter. And then that neurotransmitter binds into this little pocket on that ligand-gated ion channel. Normally, that channel has a valve that's closing it like this. When the neurotransmitter binds, it opens up the valve, if you will, and allows for negative ions to flow into the cell. These negative ions make the inside of the cell more negative than it usually is. It brings it below what's called resting membrane potential. That's called height the cell more negative than it usually is called an IPSP. Why is this involved in what's called your graded potentials? And these are basically little changes in the voltage of the cell membrane to the ability to generate action potentials. So your dendrites are involved in graded potentials. That's what I want you to know. How? Via these ligand-gated ion channels. There is another way though, and it's just important for you to remember the second way. The second way that it has other proteins here that are involved here is via what's called G protein coupled receptors. We're not going to go through this mechanism because it's long and it's unnecessary. We have other videos that cover that, but these have G protein coupled receptors that, again, imagine you have a neuron here, releases a neurotransmitter. That neurotransmitter binds onto this little receptive region of this pocket, this receptor here. When it binds onto it, it activates what's called a G protein. And that G protein can activate what's called second messengers. And these can be a bunch of different types. But eventually that activates what's called protein kinases, right? And the whole point here is that these protein kinases can go and phosphorylate particular proteins that are present on the cell membrane. And maybe this protein that's protein kinases, right? What that'll do is that'll allow for either positive ions to flow in, making a positive change, bringing about an EPSP, or bringing in negative ions into the cell. And if those negative ions flow into the cell, that could be causing a IPSP. So it's the same concept, just a different way that they get there. This is what dendrites perform this type of action, which are involved in graded potentials, whether it be ligand-gated mediated or G protein coupled mediated. Cell bodies also do that. So that means if you were to take a look and zoom in on that cell body and really look at it, everything that's going to be happening there is happening here. It's the same type of activity. So you also will have neurons which are going to be interacting here. Presynaptic neurons interacting with the cell body. Okay? So that's one thing. So one thing we already know is that this cell body is involved in graded potentials. But it has an even more significant function. Very important function. It's involved in protein synthesis. And when I talk about this, we'll go over literally the most basic aspects of what I mean by protein synthesis, the process. But what I want you to know is when we talk about protein synthesis, what type of proteins are we making? There's proteins all across this dang cell. It could be neurotransmitters that you're actually synthesizing. It could be enzymes that are involved in particular cellular processes. It could be membrane proteins. Maybe membrane proteins that are going to be voltage gated, ligand gated, or G protein coupled receptors. So it could be membrane proteins. So it is important all of that happen. We're going to synthesis. How is all of this happening? We understand the process of how it's involved in the graded potentials, which is basically designed to take resting membrane potential to a threshold potential to trigger an action potential, right? We talked about that. But how is it involved in this protein synthesis? In the basic sense here, you have the DNA inside the nucleus of the cell body, right? And that DNA may have particular genes. Maybe these proteins are gated, maybe it's for neurotransmitters, enzymes, whatever. But whenever the transcription, that mRNA is then done, it does what? It's then exported out of the nucleus and into the cytoplasm. It then comes called the rough endoplasmic reticulum. But it's important to remember that the rough endoplasmic reticulum inside of neurons is sometimes referred to as Nissl bodies. Okay, so sometimes you might hear the term Nissl bodies. And this is basically a specialized name for the rough ER in neurons. Now, this mRNA may go to this rough endoplasmic reticulum, and at the rough endoplasmic reticulum, it'll use that mRNA and then translate it. In other words, it's going to turn this into a protein. Okay? So it's going to turn that into a protein. That protein will then be packaged in the rough endoplasmic reticulum and then budded off to then be modified and further packaged by what else? By the Golgi apparatus. So then here's you're going to be consisting of the proteins. It'll then move into the Golgi apparatus. And in the Golgi apparatus, it'll undergo modification and then packaging into vesicles, where we're going to take that protein and package it inside of this vesicle. And then bud that vesicle off of what is this structure here? This structure is called your Golgi apparatus, right? So this is your Golgi. Now, from here, when you bud that off the Golgi, you bud that vesicle off, which contains in it, synthesizing here, is actually a neurotransmitter. Neurotransmitters that are
So that is a completed study session. So just wanted to say thank you so much for tuning in to a study session. We'll try to do these as many times as we can during the week, just to showcase how Kawe works. So Kawe is a web based app where you can log in and uh, create a study plan. So here I created a study plan for neuroscience, but you're also able to create study plans for other courses. So for example, I created some for immunology as well as software and biology. So if you want to create a study plan, it's pretty easy. All you have to do is go to view set and you put in the name of your course, your next exam date. Now, if you're just generally studying, it doesn't have to be just put in the name of your course, your exam date, and then the topics for your exam. So you'd put in at least four specific issues that you're facing in that class. And then once you click create schedule, it will create a study plan for you. And then for each day, it will create what are called sessions, which is one we just did. So here, you'll have sessions where you can kind of manage your studying. So just wanted to say thank you so much for tuning into our first live stream of a study session. We hope to do more of these in the future and help students when it comes to studying. So thank you so much for